Good afternoon. Welcome to our investor update call on our phase 1 ReSPECT trial for recurrent glioblastoma. As a reminder, please review our forward-looking statements language that can be found in this investor deck or our other investor-oriented presentations. My name is Mark Hedrick. I'm the President and CEO of Plus Therapeutics. I'll lead the call through our planned 1-hour agenda. First, I'll present a brief overview on Plus. Then the team will talk about recurrent glioblastoma as a disease target. And then we'll talk about the results of the ReSPECT trial thus far. And then we'll hear from a patient. Following the patient, we'll then have time for Q&A. Joining me this afternoon are Dr. Gregory Stein, Senior Vice President of Clinical Development at Plus Therapeutics. And also we're fortunate to have Dr. Andrew Brenner, Associate Professor of Medicine, Neurology, and Neurosurgery at UT Health Science Center at San Antonio. So with regard to Plus Therapeutics, our focus is on the discovering, developing, and delivering complex and innovative treatments for patients battling against rare cancers. For those of you that are new to the company, we are a Nasdaq-traded company based in Austin, Texas, with a proprietary nanoparticle drug development platform. We have 3 clinical stage oncology drugs in development, and our lead drug, RNL, is funded by the NIH and specifically the National Cancer Institutes. Our technology platform is based on our expertise in developing, manufacturing, and scaling up liposomal or nanoparticle drugs containing a variety of active pharmaceutical ingredients. Most recently, we added proprietary technology to our platform that allows us to encapsulate a variety of radionuclides in our liposomal constructs. We have 3 clinical development stage drugs, but today our plan is to focus only on our investigational drug, RNL, for recurrent glioblastoma. And so with that, I'd like to introduce Dr. Greg Stein, Senior Vice President of Clinical Development, who will present an overview on the use of RNL 186 for recurrent GBM. Dr. Stein.
Thanks, Mark. Appreciate that. So glioblastoma and recurrent glioblastoma, unfortunately, is almost universally fatal. The incidence rate is about 6.5 per 100,000, and in the US, that translates to approximately 12,000 patients a year. In the EU5, about 11,000, and probably 13,000 more broadly across the EU, and close to 5,000 patients in Japan. Nearly 100% of patients who are diagnosed with glioblastoma ultimately have recurrence, and once recurrence occurs, overall survival is measured in months, somewhere in the range of 5.6 to 10 months, depending on a variety of factors. Now, following standard of care treatment and recurrence, the treatment options for the patients is somewhat less well-defined. There is the opportunity to have a second surgery. The patient can be retreated with radiation. They can be treated again with temozolomide. Those 3 therapies really form the core of the initial upfront therapy. They can also be treated with bevacizumab or other drugs such as lomustine. These drugs do provide some palliation, but in general provide very limited benefit in terms of overall survival. And because of this, there is a significant unmet need in recurrent glioblastoma for an effective therapy. Now, briefly, RNL, or Rhenium 186 Nanoliposome, has 3 core components and a unique delivery system. At the heart is Rhenium 186. This is a radionuclide that is a beta and gamma energy emitter. 90% of the energy is beta energy, And these are high energy electrons that travel on average in tissue about 1 to 2 millimeters in distance. So we can control very tightly where the radiation energy goes. And the Rhenium-186 has a half-life of 90 hours, meaning that within, over the course of a couple weeks, most of the activity has decayed. Now, Rhenium-186 is bound to a molecule called BMEDA, and BMEDA allows us to secure the Rhenium-186 in a small 100 nanometer liposome, which is stable and retains the Rhenium-186. And we deliver this via a method called convection-enhanced delivery. And this is simply a catheter that is inserted by a neurosurgeon directly into the tumor mass or very close to it, and then using a hydrostatic pressure wave, the drug is dispersed throughout the tumor and surrounding tissue as needed. Now, the core value proposition for RNL really covers two key aspects, but the retention of the drug in the tumor and the effective dispersion of the drug throughout the tumor. Now, if we look here at the graph and these photos down here, rhenium that is not in a liposome quickly clears, here's time, from the tissue and is very highly concentrated. Once we put it in a nanoliposome, it stays for a prolonged period of time, and we're able to much more easily get good dispersion throughout the tumor. And these are from rat models here. Now, preclinically, we see a survival benefit in both U87 and U251 models. And in the pictures here, we can see the tumor going away and actual ablation of the tumor over time, whereas in the control, the tumor continues to grow. RNL preclinically has also been shown to be quite safe. In a GLP toxicology study, doses up to 6 millicuries were administered, and the absorbed dose was 360 gray, which is about 10 times what is typically given in a recurrent setting for humans. And at that dose, we saw no adverse effect limit, and this was the dose that was taken into the clinical trial. So very briefly, the clinical plan is one in which we preoperatively take the patient's scan, and the neurosurgeon and Dr. Brenner plan out where the catheters will be placed, and there is very complex software that allows them to do this. The patient's then admitted to the hospital, and for us, this becomes day one. The neurosurgeon, using stereotactic surgery equipment, drills a very small hole in the patient's skull and advances the catheters into the tumor. These are secured, and the patient then returns to their room. The following day, the patient then is transferred down to the nuclear medicine department, where RNL is delivered by convection-enhanced delivery, and in real time, we can visualize that delivery. And 2 days later, the patient is discharged home and able then to continue on with their normal activities. And with that, I'll turn it back over to you, Mark.
Thanks, Greg. So now I'd like to take the time to introduce Dr. Andrew Brenner. Dr. Brenner, as I mentioned, is the principal investigator of the ReSPECT trial and associate professor at UT San Antonio. Dr. Brenner? Dr. Brenner, are you still with us? Dr. Brenner? Chantel, the operator, could you mind just checking if you still see Dr. Brenner's image on the on your?
For some reason, I froze up.
Oh, there he is. Hey. Hey, Dr. Brenner, can you hear me okay?
The app. Yeah, I can hear you fine. Sorry, it froze up just for a second there.
Okay, I was getting ready to tell a Texas A&M joke. So good thing I didn't have to do that. So I've introduced you, and please kick it off whenever you're ready.
Yeah, let me just get the slides back up here because it's not showing. I'm having to.
No worries.
Which slide number are we on? We're on. Get back to it here.
17, I believe.
Okay, now if I could see the slide.
Yep. It's titled "ReSPECT U.S. Phase 1 Clinical Trial."
Yeah, advanced. It's not showing on mine, but I'll just have them here. So the clinical trial was designed as a multi-center study with open label, so everybody knew what they were getting. And it's an escalation in both the volume of administering as well as the dose. And the idea is to find the safety, the tolerability, and the distribution of the drug using this convection-enhanced delivery technique that Dr. Stein talked about a moment ago. And the patient population is a recurrent glioblastoma patient population after they've already received standard surgery, radiation, and chemotherapy. Study design, again, is a prospective study. We use something called a modified Fibonacci scheme, where we're doubling the dose, and then we increase in fractions thereafter. And the idea is to find the recommended phase 2 dose, the dose we're going to evaluate more broadly. We plan for up to 55 patients, and the 55 patients includes both the dose escalation part as well as a planned expansion or a phase 2 portion where we're looking at activity. The 5th cohort has just recently been completed. We're now in the 6th cohort. And I'll move on to the next slide here. So this shows.
I'll advance for you, Andrew. So just go ahead and just give me the heads up and I'll advance.
So in terms of dose escalation, this is showing what the different dose levels we're looking at. We are currently at the cohort 6, which we're infusing a dose of 22.3 millicuries in a volume of 8.8 milliliters. I'm showing the data today up to 13.4 millicuries in a volume of 5.28 milliliters. Next slide. This is a summary of the patients and shows what we've seen both in terms of outcomes as well as the patients, what they received. We started at a dose of 1 millicurie in a volume of 0.6 mmLs. So a very small volume, started with a single catheter and a low flow rate. And we have subsequently, with each cohort, increased the dose that we administered by dose doubling. And now we're in this fractionation portion. So it went from 1 to 2 to 4 to 8 and then at 13.4. And the volume went up as well to the 5.28. We increased the number of catheters, starting with 1 catheter to make sure that we could describe the distribution of the drug. And now we're at 3 catheters. And we've also increased the infusion rate, which is important both for the timing of the procedure to shorten the length of the procedure, as well as to increase the volume of treatment. With higher rates, we can get a higher treatment volume. And you can see here some encouraging signs for survival, and I'll show more data on that in a moment. Next slide, please. So this shows the safety data from the patients treated through cohort 5. It's important to note here that, as indicated below, that neither the incidence nor the severity of the adverse effects appear to increase with increasing doses of RNL. So that's a very promising signal that with increasing doses, we're not seeing increasing toxicity, which means this is a safe drug. And most of the A's were considered to be causally unrelated to Rhenium NanoLiposomes, with the exception of scalp discomfort, which is an expected potential side effect from having the catheters placed surgically. Next slide. So as we went up on the volume, we went up on the size of the tumors we were treating. We did not want to treat larger tumors which could not be adequately covered by the therapy. And you can see here that with each cohort, the size of tumors went up. And we are now routinely treating patients with tumors between 15 and 20 milliliters, which corresponds to almost a 3-centimeter tumor. And when we look at the doses that were achieved to the tumor volume, we see that we are getting now in the most recent cohorts routinely between 300 and just over 500 gray of absorbed dose. And it's important to note that in the recurrent setting by external beam radiation, we typically only use doses around 30 gray. So anywhere from 15 to 20-fold the customary external beam dose that you would give. The median absorbed dose to the tumor volume across all subjects was 15. In patients who failed bevacizumab, it was much lower, 38 gray. And we think this is due to the effect of the bevacizumab on the convection volume, that it actually interferes with the convection, and when there's some biology behind that. For patients who were bevacizumab naive, the dose that was achieved to the tumor volume was around 367 gray, and the maximum dose to the tumor volume was 593 gray. Next slide, please. This just shows the absorbed doses in the tumor compared to the brain as a whole, as well as the entire body. And so in green there is to the tumor volume, and you can see with the most recent cohort that we're reporting, 13.4 millicurie cohort, cohort 5, patients had a total absorbed dose to the tumor volume of 423 gray, whereas their surrounding brain only received a total dose of less than 1 gray, 0.4 gray, and to the whole body, it was very negligible at 0.1 gray. These are insignificant doses. And the ratio of the dose to the tumor volume compared to the body is greater than 3,000. Next slide. This gives you a idea of the delivery of the Rhenium NanoLiposome. So in the top row there, you see the patient's MRI. In the first column is the axial plane, the middle column sagittal plane, and the last column in the coronal plane. And you can see 24 hours after the infusion, you see the distribution in the middle row of the Rhenium NanoLiposomes based on SPECT-CT imaging. And you can see on the MRI above it, we have the outline of that to correspond to the area that was treated. And then when you look at 120 hours, we see excellent retention. All of the radioactivity remains within the tumor volume. So excellent coverage as well as retention. And in this case, the tumor volume was 6.5 mLs, and the coverage was greater than 90% with an absorbed dose to the tumor volume of nearly 420 gray. Next slide, please. So this shows one of our patients over time. Again, this is in the axial plane, and you can see that patient who had excellent coverage. His tumor initially day -5, which is 5 days before the treatment. The second image is day 28, then day 118, and then day 362. And so over a year out, we're seeing some decrease in the enhancement or the tumor volume. And the patient remains alive and is nearly 2 years out from his initial treatment. Next slide. This is one of our more recent patients, and you can see on top the image of the tumor, the large enhancing volume there. Again, axial on the first, sagittal on the second, and the coronal in the third column. And then 24 hours later, we see the absorbed dose to the tumor. And at 120 hours in the bottom row, you can see it's actually spread out a little bit even. We even get better coverage at 120 hours. So excellent retention, good tumor coverage. The tumor volume in this case was higher than that last patient I showed you and was almost 19 milliliters, and the coverage was 87%. The amount that was absorbed to the tumor was 336 gray. And at 24 hours, even the most anterior portion that we were maybe not covering was covered as it redistributed, and the entire tumor was in the distribution in the purple line in the top figure there. Next slide. This shows the effect. And so you see in the first column before treatment and day 56 on the second column, and you see the loss of all that white there, which is the enhancement and corresponds to tumor. We know that that's a loss of tumor because in the figure below it, you see what we call as an RCBV map, which shows the perfusion or the blood vessel supply. And so we see a loss of that perfusion in that image below. And then in another image in the second grouping there, you see the enhancing tumor in the third column and the loss of that enhancement in the fourth column. And again, associated below that with a increased RCBV or increased blood flow to the tumor pre-treatment and a complete loss of that blood flow post-treatment at day 56. Next slide. This is a review of the survival to date, looking at the patients who had not received bevacizumab and who have received bevacizumab. And it's important to note that for patients who fail bevacizumab, their survival at that point is significantly shorter and ranges from 90 to 120 days. The typical survival for patients who have not received bevacizumab yet is about 8.3 months, depending on what studies we look at. And we can see some very encouraging signs in terms of survival in these patients. The patients indicated by the green circles are patients such as our patient here today who has been recently treated and continue to be followed for their survival and who will continue to do very well. But we do have some encouraging signs here. The median survival duration of subjects that had failed bevacizumab was 4.8 months, which is just a little bit above what we see for the general population. And then median and mean survival duration in subjects that were bevacizumab naive was 11 months and 15.4 months, with 4 of our patients still recently ongoing and recently treated. Next slide. So with this, the conclusion is that we can deliver up to 15 times, and maybe even if we consider the recurrent doses, up to 20 times absorbed doses of radiation administered by external beam radiation therapy, and we're not seeing significant evidence of toxicity. It's also important to note that this is administered within a few days, whereas external beam radiation is administered over weeks. These data indicate that intratumoral administration of RNL by CED at doses up to 13.4 millicuries is safe and well tolerated. No treatment-related adverse effects noted thus far, and maximum tolerated dose not yet reached. Prior administration of BEV seems to negatively impact convection of RNL. And in the subsequent survival study, in the phase 2 study, we do not plan to enroll patients who have failed bevacizumab. And that's pretty standard for phase 2 studies. And the current status, we are on cohort 6. We've recently administered 22.3 millicuries and 8.8 mLs. And we have 2 more patients upcoming in the upcoming 2 weeks. With that, I'll stop.
Thank you, Dr. Brenner. So yesterday, I actually had the opportunity to speak with Phyllis, who is a ReSPECT clinical trial patient. She's one of the 2 patients that have been most recently treated that Dr. Brenner had referred to previously. She's had 2 previous surgeries prior to getting into the trial, chemo and radiation. And I felt like it was good to get her to talk about her experience with RNL as it related to the other treatments she had had. So I'd like to just let her talk in her own words. There we are. Hi, Phyllis.
Hi.
Are you able to hear me and see me okay?
Yes.
Okay.
Hello.
Good. Well, hey, thank you for taking the time. And you are currently a patient that's participating in the ReSPECT clinical trial for RNL-186 for patients with recurrent glioblastoma. So first of all, I just want to thank you for participating in the trial and then agreeing to just talk for a few minutes.
Okay. You're welcome.
And how are you doing generally?
I'm hanging in there. I haven't had any major side effects that have caused me to be incapacitated.
Yeah. Well, you look good from here. So Phyllis, I've got a couple of questions for you that I thought might be a good way to kind of assess the how things are going with you with respect to the treatment. So maybe kind of going backwards a little bit, would you mind telling us a little bit about how you found out that you were sick, and then, how the treatments and the overall experience that you've had thus far?
Sure. So my initial diagnosis was through a seizure. I was actually working as a veterinarian technician, which was my career. And I went into work that day, not really knowing what was going on with me physically. I was kind of out of it. And it happened to be a surgery day. So my coworkers noticed that I wasn't myself and brought me into the surgical unit and kind of talked to me and said, something's not right with you. So they called my husband, and he actually came and picked me up, and we went to the medical center. And that's where they did the first scan and saw something suspicious. So then transferred me to a different hospital by ambulance. And then that's where I stayed, and that's where I was diagnosed with suspicious growth, but not diagnosed at the time. So we went ahead and did the first surgery. And at that time was diagnosed with the astrocytoma grade 2. And it looked like we had got it all. Unfortunately, we know now that wasn't the case. So from there, I did the at-home Timidar radiation treatment, went through the 30 days of the recommended treatment. And then in August found out that the tumor wasn't responding. It actually had grown quite a bit. So then that's where Dr. Floyd, my surgeon, came in to the case, which Dr. Brenner was already on the case with me. So then Dr. Floyd agreed to go in and basically kind of redo the section and kind of try to give us a fresh start. And then at that time, I went on a study of it's a long name, but it's SASE. You probably have some history there. That was used in case where breast cancer metastasized. They were finding they were having some results when it went to the brain. So this was a new study that we started. I had the first treatment basically the night before my surgery, and did that for several months. Then found out I wasn't responding to that anymore. So that's when the next study was offered to me with catheters, with radiation straight into the tumor. So
it's like you've had a couple of surgeries so far before you even got into the study.
Correct. This last one, yeah.
So, so you found out about the trial with the catheters and the radiation, the RNL trial through Dr. Floyd and Dr. Brenner?
Correct.
So how did you find that treatment experience with the RNL? That's a little different than having surgery, I guess. And
yeah, it was, you know, I had it right there at my birthday. So it was a little scary, but so I found it a lot easier than actually having surgery. I recovered really well. It was a low pain level. Mm-hmm. So they said I was one of the best patients. So I took pride in that.
Well, that's great. And you were in the hospital for a couple of days around the time that you had the catheters and the infusion?
Correct. So I think I went in on the 14th and actually had it on the 15th, which was my birthday, so that's easy to remember. Uh-huh. So they did the actual placement of the rhenium and stuff on my birthday, and then took all the pictures and everything. And then I had to, once I got discharged, I came back every day and got like a scan to make sure that we were getting good absorption, but it wasn't leaking out. So they were very happy with how much they were able to get in there and how much it retained. Mm-hmm. So I would say I'm a success story so far.
Look good to me from here. So whatever you're doing, keep doing it.
Yeah.
How was the RNL treatment kind of versus radiation? It sounds like you had a about a month-long course of radiation?
Mm-hmm. You know, the radiation, it does make you really tired, so that was probably the toughest part. And then as far as side effects afterwards, there was some side effects like just forgetting things, and of course I've had to be on kind of a high dose of steroids to keep the brain swelling down. So that would probably be my thing that I've disliked the most, is the side effects from the steroids. But you know, nothing that I would call major, like where I can't function, or, you know, there were some things that I would have liked to have done without, but, you know, things have side effects. That's just part of most medical procedures, I think.
Did the catheters bother you when they took them out? And was the infusion difficult for you, or?
No, I mean, you know, it was more patience. I felt like the center of attention. I was everybody's center of attention with the doctor. So, yeah, I mean, it was kind of a slow process. I was in there several hours, but that was probably the most trying time, was just the patience of laying there while they did all that.
They were infusing it into the catheters, right? The RNA.
But I think I missed some of the trial and error with that they had gone through with prior patients, so I was kind of at an advantage there.
So would you recommend this to other patients? Let's say that other patients that might be interested in the trial, or, you know, maybe one day, hopefully, if this gets approved by the FDA, would you recommend that treatment to other patients?
I would. I mean, I think I think it was, you know, successful, seeing as my tumor is so far stable and not growing crazy like we know they can. So I'm very happy about that.
Yeah, great. Well, look, I don't want to take up too much of your time, but I really appreciate it. Anything else you'd like to say or tell us regarding your experience as a patient with the RNL 186?
I just think it's good that, you know, if people can volunteer, you know, it's all got to be a case by case, but I think this is how we sometimes get somewhere further in our studies, is, you know, I don't look at myself as anybody special. I'm just trying to maybe help another human. Mm-hmm. And hopefully this will do that.
Well, Phyllis, thank you very much, and look forward to talking to you again. And obviously, if anything we can do to help, please let us know. And take care.
Okay.
Keep up the great work. You look great, and it sounds like everything's going really well.
Well, thank you, Mark. Have a good rest of your day.
You too. Take care.
Okay. Bye.
That was Phyllis. So let's close out the presentation, and then let's go to Q&A. There are a number of questions that are in the queue. So, in terms of forthcoming milestones for 2020 and 2021, our plan is to complete enrollment in the U.S. ReSPECT Phase 1 clinical trial, report the final ReSPECT data, and determine a Phase 2 pivotal dose recommendation. We potentially will seek US FDA breakthrough therapy designation and European orphan drug designation for RNL in RGBM. We also intend to complete the key CMC and GMP commercial scale-up activities for manufacturing of RNL, and also optimize the regulatory strategy and follow-on clinical trial plan for RNL as it relates to GBM. We also intend to complete IND-enabling studies for the first follow-on RNL indication beyond GBM and explore CPRIT support for ongoing RNL development. And then we'll continue to explore partnering support, not only for RNL, but for our other 2 clinical stage assets. So with that, Chantel, I think you have something you'd like to read to potentially provide guidance on how to ask questions.
If you would like to ask a question through the webcast, please use the Q&A box on the webcast console. To ask an audio question, you'll need to press star 1 on your telephone. To withdraw your audio question, please push the pound or hash key. Please stand by while we compile the Q&A roster.
So let me just read the first question. So the first question is about the use of steroids and requirement for steroids in RNL treatment or just standard radiation. Dr. Brenner, what is how do you use steroids in this trial?
So in order to not mass toxicity, we do not pre-treat with any steroids. Investigators are encouraged to use them if needed after the treatment. We haven't had any marked steroid needs. We have had patients that have intermittently required steroids. Some patients with GBM have chronic steroid needs, and that just is the nature of their tumor. And so we have had patients that have had to continue on their steroids. We have had patients that have had to temporarily increase their steroids, but so far we haven't seen any dramatic increase in edema associated with the treatment.
Great. The next question is about the RNL. How long does it stay in the brain? And can you talk about, Dr. Brenner, the adverse effects on normal brain tissue and what you've observed thus far in these patients, and kind of how does that compare with external beam radiation?
So the rhenium just decays, and as mentioned by Dr. Stein earlier, it does decay over a period of a couple of weeks. The only thing that really remains, number one, the liposomes are made of material that the same cell membranes are made of, so it just becomes inconsequential. And then the BMEDA is present in very small amounts. The amounts that we use are considered trace amounts in order to trap the rhenium. And it's a stable molecule that is relatively inert, so we don't see any significant toxicity from that. In terms of the normal brain, first of all, the path length is very short, so this only goes where we put it. So as opposed to external beam radiation, photons have to pass through the surrounding normal brain in order to reach their target. We use things like tomotherapy, which is really innumerable number of beams. As it spins around, we modify the beam shape so that there is some conformity to it, but ultimately the photons still have to pass through the normal brain, and that's why you have a very narrow therapeutic window when it comes to external beam radiation, whereas these beta particles only have a path length of a couple of millimeters. They go where we put them. There is some radiobiology published in terms of beta emitters toxicity versus photons. It is different. I don't claim to be a radiobiologist. I'm not going to go too far into that, but suffice it to say that that for normal cells, they should be able to tolerate dramatically higher doses. Number one, because they should be quiescent and not replicating. And number two, they have their checkpoints in place. So DNA repair pathways remain in place in those cells where they're abrogated in the tumor cells. So it's expected that similar to radioactive iodine therapy, where we can give hundreds of gray of dose for thyroid cancer without seeing toxicity to the surrounding neck, that this would be very tolerable for the surrounding normal tissues.
The next question, also for you, Dr. Brenner, is what is the median follow-up time in the 15 patients, as well as stratified by Bev experience? In other words, sort of naive versus Bev recurrent. Dr. Brenner, are you still with us? Brenner's not with us. We'll save that question in case he dials back in. Next question for you, Dr. Stein. What was the tumor coverage percentage in the Bev naive versus the Bev pretreated patients?
Good question. So on average in the Bev naive patients, and there were in the Bev treated patients, there were 7 of those. And on average, we only got about 40% tumor coverage. In the Bev naive patients, across all 8, it was about 80%. And in the last few patients, it's been around 90%. Some of the early ones, it was such small volume that it was difficult to get full coverage, but clearly a significant difference between those who had been pre-treated with bevacizumab and those who had not. And then I can maybe, unless Dr. Brenner's back on, I can.
Sorry, my browser froze up again.
Go ahead, Greg, or Dr. Brenner, go ahead if you're back on. And would you like me to read the question again?
What was the question, Mark?
The question was, what's the median follow-up time in the 15 patients, as well as stratified by bevacizumab experience? In other words, naive versus beva recurrent.
The cohort first, so we're probably, some of those patients, as you can see, are well beyond 30 months. Of the ones that are active, those were the green circles. We do have 3 patients that are currently within the last 4 months. And we have, I think, 2 that are out past 1.5, 2 years. So I can't recall what the median follow-up is, but it's probably going to be a pretty big range because we just started pushing accrual again recently.
There we are. Hi, Phyllis.
Hi.
Oh,
Are you
sorry,
able
guys.
to hear me and see me okay?
All right.
There's
We stop
Phyllis.
that.
Yep.
And the question was
There you
in
go.
terms of the Bev. In terms of the Bev naive patients, we pretty much stopped accruing them after cohort 4. There were no further Bev refractory patients after cohort 4. So cohort 5 was completely Bev naive, and then the first cohort was completely Bev naive as well.
So, Dr. Brenner, another related question. What historical survival range for beva failed recurrent and beva naive recurrence are you using? Is the data thus far in the range of survival data in these settings that has been shown with beva and or beva plus chemo? I think you sort of partially addressed that, but would you elaborate further on that?
So there are multiple studies in terms of the survival both for bev naive and bev failure. We recently published as part of the phase 2 study of BB111, we went back and did a meta-analysis of all the studies to look at survival for patients who went on subsequent studies, and it was around 8.3 months. So for bev naive patients, it's 8.3 months. For a second bev, for patients who failed bevacizumab and went on a second regimen, if they did not receive bevacizumab, it was relatively short. It was only about 2 to 3 months. And if they maintained on bevacizumab, it was 3 to 4 months. So not much difference there, but some slight difference if they continue on bevacizumab.
Okay, great. We've got 3 questions from the same questioner. Question 1 is, how do these results compare to standard of care?
Well, so let's talk about standard of care real briefly. So for standard of care, we don't have really any FDA-approved options except for really Avastin. I mean, we do use things like Avastin plus lomustine, PCV, irinotecan, carboplatin, those things do not have any proven survival benefit. So we really have nothing with proven survival benefit after recurrence from chemoradiation and tumor treatment fields. Once those three modalities are given, it's kind of physician's choice, and nothing really has been proven to make a difference. So there really is kind of no well-established standard of care that improves survival as of yet. So it's really too early to make a efficacy analysis with a handful of patients. I think what I would point at is that from the few patients we have that are bevacizumab naive, it's very encouraging. And we need to go and do additional studies to look at a larger number of patients, and that's our intent. The other thing that's really important to make mention of in this study is we are only just now getting into the doses that we really want to give, where we're giving doses large enough to cover reasonable sized tumors. So, this next couple cohorts, or this expanding this cohort and looking at the patients that we're now treating in the next few weeks is going to be very exciting, as well as important for us in terms of where we are with the recommended phase 2 dose.
Second question from this questioner is, how would you expect RNL therapy to perform if patients are treated earlier in their course?
Well, obviously earlier is better, and there's a couple different indications where we are thinking about. Number one is for patients with biopsy-only disease who are not able to undergo debulking. This could potentially be an could be a therapy where you can augment external beam radiation. So, and those patients are typically diagnosed with biopsy, dropping a catheter at the time of that diagnosis, and treating them right away, getting their treatment done within a couple days before they even get to external beam radiation would be very attractive. The other patient population is, if you think about the MGMT unmethylated patients, those patients don't do well long-term, and they get no benefit from temozolomide. So, so obviously it would be attractive to do that. First, we need to get to the recommended phase 2 dose, know what our phase 2 dose is, and expand in terms of the current setting. Then we'll know better how to move forward into the newly diagnosed setting, but there certainly would be some enthusiasm for looking earlier in the course of the disease.
Okay, Andrew, again for you. Although the data seem early and we're still in dose escalation, however, can you tell what number of month survival? In other words, what's the length of survival that would be meaningful in your mind, and how does the quality of life matter as Bev has some side effects in the recurrent setting?
Yeah, that's a great question, and I think Phyllis hit this a little bit, but you have to remember when you're getting the initial radiation therapy that you're going for radiation every day for 6 straight weeks, Monday through Friday. This is given in 3 days and you're done, and you really kind of just sit in the hospital for a few days, which is the biggest negative, is that you have to be there for a few days. But it's very tolerable in terms of that, at least so far. And then when you think about other therapies, a lot of times you don't give just bevacizumab. You give Bev, plus you give irinotecan, or you give lomustine, et cetera. And those are associated with significant toxicities. And we do that really to try and keep the disease at bay, but without any real promise of survival advantage for the average patient, at least. Some patients might, but on average, it doesn't increase survival. So what's meaningful? I think that's up to that. There's maybe higher powers than us, but I think if we told most patients that a 2 or 3 day procedure would improve their survival by 2 months, would they take it? I'd say the vast majority would. Ideally, I think what you would hope is that you would get some longer term gains, and we are seeing that with a few patients. It remains to be seen when we get to a larger number of patients, but I'd say that, at least a 20 to 25% improvement in survival would be something that we'd want to go to in terms of goalposts. For the phase 2 that we have, as it's designed right now, that's kind of what we're looking at, is a improvement to a little over 10 months but we still have to determine where we are with in terms of the recommended phase 2 dose before making hard decisions on the subsequent trial.
Great. So next question, I'll give this to Dr. Stein. What are the next steps for the phase 1 enrollment, phase 2, or other clinical trial plan?
Thanks, Mark. So we need to complete cohort 6, and that would be 2 more patients, and then we'll evaluate the data and see where we're at. The ultimate decision at that point, we have to go to the Data Safety Monitoring Board if we're considering a further dose escalation, which would allow us to potentially treat larger tumors. Although at this point, we can treat a tumor that's probably about the size of a golf ball. So that's a good number of the tumors that patients will typically present with. And the real goal at this point is to develop some confidence in that recommended phase 2 dose and feel like we are comfortable with the number of catheters, we have the right flow rate, we are confident in the size of the tumors we can treat, and then from there we'll take that into our phase 2 pivotal trial design.
Dr. Brenner, anything to add to that?
No, I think that summarizes it well. I mean, I think before we make any hard decisions, we really have to learn a little bit more in terms of both the delivery and the tolerability of higher doses so that we can maximize the potential benefit for patients when we get into a phase 2 trial.
So 2 more questions, and that ought to get us right at an hour. One question, I guess, for you, Dr. Brenner, is these patients are getting a significant amount of radiation injected directly into the body. What are the precautions that these patients need to adhere to, or their family members or caregivers? We need to put them in isolation, or how is that handled clinically?
Yeah, that's a really good question, and this goes back to the safety of beta emitters. There's a couple different points to this. First, what we know from all the pharmacokinetics that we've done so far is that whatever is not trapped is rapidly cleared from the system. So the liposomes get retained within the tumor. Anything that doesn't get retained gets picked up, the liposomes get processed, and the rhenium gets dumped. Rhenium gets really quickly cleared from the kidneys, so we see almost complete clearance of any radioactivity that's going to be left behind that's not going to retain within the tissue is cleared within a couple of days. Matter of fact, we stopped even looking beyond 2 days because in all the patients we treated so far, all of the radioactivity was eliminated through the urine within that first 2 days. So there's really no precautions. The radiation can't make it all the way through the skull, and the urine is basically clean after they leave the hospital. So there's really not much to look at or to be concerned about or precautions that need to be used thereafter.
Great. There are a couple more questions, and I think probably in the interest of time, we'll cut it off with these last 2. So the question is, how long does RNL need to start showing effects? Presumably, those are imaging-related effects. Slide 24 suggests that the tumor gets larger before it starts getting smaller. Dr. Brenner, can you address that?
Yeah, that's an excellent question, actually. So we have seen both. We've seen cases where enhancement just quickly disappears, perfusion gets, is gone, and we see that within 2 months, as in one case. Whereas in other cases, we do see some evidence of potential what we call pseudoprogression, where it looks a little bit worse at first, and there might be some edema, and then it kind of gets better over time. So we haven't fully characterized this yet. This is where having more patients will better let us understand what to expect in terms of which patients might have some immediate response and some patients might have some pseudoprogression. There's a lot to learn still, but both of those seem to still be possibilities based on what we're seeing.
Great. So I'll make this the last question. So we've got about five or so minutes, so we can end right on time. But the question is about surgical planning, the accuracy of catheter placement, and the ability to obliterate the tumor that is enhancing. But how do you place the catheters in such a way that you could also target the non-enhancing tumor that might be responsible for recurrences?
Yeah, we're very fortunate to be in a time right now where we have excellent convection-based tools. We use the Brain Lab system, which includes the iPlane Flow software, together with the VarioGuide system and a flexible catheter. That system has great accuracy. The main limitation is how you co-register intraoperatively. And we have seen that there is maybe some variability there, especially when you look at the posterior tumors. There's some things we can do. We can use fiducials, which are some things that we can attach to the skull itself, so that when we get into the operating room, we have maximum precision. But we're able to get pretty darn accurate with the fusion techniques we use, even without the fiducials. In terms of where we place the catheters, this is a lot of, it depends when you talk to the community, there's different opinions about where the catheter should be put. Some people say, let's drop it right in the middle of the tumor. We've kind of taken more of the approach that we want to place it more in the leading edge of the enhancement where the area of infiltrating tumor exists. And so that's what we've been doing. When we do that, we are actually, in terms of looking at the volumetrics, when we talk about coverage of the tumor, we're talking about the enhancing portion of the tumor, because per RANO criteria right now in a bevacizumab naive patient, the enhancing component of the tumor is what is used for response. That said, we do try and cover the non-enhancing as much as possible. And now that we're getting into these much higher volumes that we're using, because we keep escalating the volume we administer, and we're also increasing the rate at which we administer it, that the volume that we're able to cover is more, so we can make sure to cover more of the non-enhancing tumor as well. So hopefully we'll see even better benefit as we get to do that with higher volumes.
Great. Well, I think we're sort of at the end of our time. For those of you that are watching this live, or those of you who may watch this in a recorded format, I hope this has been instructive, and I hope that it's brought you up to a higher level of understanding with respect to RNL for recurrent glioblastoma. I'd like to thank particularly Dr. Brenner for being with us and being available to answer questions. Also, Dr. Stein. Perhaps most importantly, I'd like to thank the patients and the doctors and support personnel that trust us in executing this trial. And then, of course, I'd like to thank our employees and then again, you guys that have all participated on this call. So thank you very much. Our contact information is here on the last slide of the deck, and feel free to reach out if we can answer any questions. And beyond that, have a good evening. Chantel, I think we're finished.
Hey guys, we're back on the private line if you need anything.
Good. Yeah, I think I figured out what my problem was. I had a PowerPoint open in another tab, and I think it was sucking memory, so it kept freezing.